Hello, everybody. Today I will talk about our work on the development of the herbicide-resistant Camelina Cultivar SE1154Hr, which was a collaborative effort between Smart Earth Camelina Corporation, Agriculture and Agri-Food Canada, and the University of Alberta. As most of you are familiar with Camelina, I will get right into the weeds. The development of a herbicide-resistant Camelina cultivar has been a focus of our work for quite some time because, for one, Camelina is a poor competitor with weeds early in the season, and secondly, the current weed control options in Camelina are very limited. As of today in Canada, the active ingredient quizalofop is registered for the control of grasses and glyphosate for pre-harvest weed control. Camelina is indeed tolerant to dinitroaniline herbicides, and as of last year, ethylchloroline is registered for pre-emergence weed control. Other than that, and particularly after emergence, Camelina is susceptible to pretty well every other broadleaf herbicide. My colleague Eric Johnson has done a lot of screenings of different broadleaf herbicides, and this is only a small sample. But all that he could find was a variety of solutions to control Camelina volunteers. Among the different herbicide classes to target for the development of herbicide resistance in Camelina, the group 2 herbicides came up as the obvious choice in a not so obvious way. But first of all, how do group 2 herbicides, also known as ALS, or AHAS inhibitors work? ALS or AHAS is an enzyme that in plants and microorganisms catalyzes the first reaction in a common pathway that leads to the synthesis of branched chain amino acids. ALS or AHAS inhibiting herbicides inhibit the AHAS enzyme by blocking substrate access to the active site, and by doing so, starve affected plants of branched chain amino acids, resulting in a variety of symptoms, including death. However, there are a number of point mutations in AHAS genes, either occurring naturally or induced through mutagenesis, that prevent AHAS inhibitor herbicides from binding to the active site and make plants more or less resistant. Group 2 herbicide resistance in Camelina is an obvious choice because, for one, it's fairly easy to create through seed mutagenesis using EMS. Group 2 herbicides also have a number of advantages in terms of their efficacy, ease of use, and low environmental impact. However, because there are a number of point mutations in the AHAS gene that confer resistance, and because group 2 are such a popular class of herbicides, there are already several weed species that have evolved to be resistant to one or more group 2 herbicide. And that has called the usefulness of group 2 herbicides into question. On the other hand, however, Camelina is extremely susceptible to group 2 carryover in the soil. And that in Western Canada actually significantly reduces the number of acres that are available for Camelina production. Group 2 herbicide resistance in Camelina would alleviate the current recropping restrictions, and also, and the usefulness of that should not be underestimated, would control Roundup Ready and Liberty Link canola volunteers. The team of Linda Hurl at the University of Alberta mutagenized Camelina seed, and after comprehensive screenings in the field and in the greenhouse, we identified 4 mutant lines with superior resistance to a combined application of thiophansulforon and tribanuron, which are the 2 active ingredients in the product Refine SG. The sequencing of the AHAS genes in these lines showed that we were dealing with AHAS mutations in all 3 subgenomes, which was very, very surprising. What was also very interesting is that in all these lines, the mutation in the AHAS genes was always a SNP at the position 580 in the gene, which led to a codon change. And that codon change resulted in an amino acid substitution in the resulting enzyme from proline to serine at position 194. And that position is homologous to the well-characterized Pro197 mutation in Arabidopsis. While all this was very exciting, what we also had to learn in our first field trials was that one mutated gene does not confer commercially acceptable levels of resistance to thiophansulforan and trabenuran, which would be resistance to 2 times the recommended field application rate. Because of that, we conducted crosses between all the mutant lines and developed 5 lines that were homozygous for different combinations of 2 resistant AHAS genes. And these 5 lines, plus the original control, were grown in the field again and tested for tolerance to a total of 7 different group 2 herbicides. One line that stood out in this trial was a line that was derived from a cross of parents that carried the resistant AHAS variants in subgenomes 3 and 1. And this line showed the highest levels of tolerance to thiophansulforan, which is the active ingredient of Pinnacle SG or Harmony SG in the US. In those response trials, we compared the level of resistance in the double mutant, here shown in green, to that in the respective single mutant parents, shown in red and black, and to that in the wild type, shown in blue, to an application of thiophansulforan ranging from 0 to 81 times the recommended field application rate. And the results of this study not only showed that the double mutant is about 1,000 times more resistant than the wild type to thiophansulforan, it also showed that the one single mutant, CS-AHAS1, was more resistant than the other, CS-AHAS3, which we think may be due to differences in the levels of gene expression, which remains to be verified. Field evaluation of the double mutant at 0, 1, 2, and 4 times the recommended field application rate of thiophansulforan showed that there was no reduction in seed yields in the 1 and 2x treatments compared to the untreated control at locations where no other stressors were present, such as downy mildew. As you can see from the labeling in this picture, the double mutant line was at that time, in 2016, considered a plant with a novel trait, a PNT. And that meant that we had to grow this line in Canada only in strictly regulated and controlled, confined research field trials. As Gerhard Rakow, a well-known Canadian canola breeder, used to say, farmers grew crops, not traits. So the next step for us was to bring the herbicide resistance into a Camelina cultivar with superior agronomics and seed quality. And for that, F4 plants homozygous for both resistant AHAS variants were crossed with the large seeded Camelina cultivar SES0787LS, also called Cypress, followed by 4 rounds of back crossing with SES0787LS as the recurrent parent. After completion of the 4th back cross, plants were salved for 3 cycles to reach homozygosity at both resistance loci, which resulted in the development of the herbicide-resistant Camelina cultivar SES1154Hr. Field evaluation of SES1154Hr after application of thiophansulforan at a 1 and 2x rate in comparison to its herbicide-susceptible parent cultivar SES0787LS showed that there was no measurable herbicide damage, no reductions in seed yield, and no delay in flowering and maturity after herbicide application compared to the untreated control. In contrast, in variety SES 0787LS, we observed yield reductions by half with every increase in application rate and significant delays in days to flowering and days to maturity. As we had expected, the seed quality of SES 1154HR was similar to that in SES 0787LS, and for both varieties, the seed quality was not affected by a herbicide application. A picture says more than 1,000 words, they say. So here's a picture of the herbicide susceptible parent cultivar SES 0787LS, untreated and 14 days after an application of thiophansulforon. And here is a picture of the same trial, but plots of the herbicide-resistant cultivar SES 1154HR, also 14 days after application of thiophansulforon. Despite the high degree of sequence similarity between the three AHAS orthologs in Camelina, we were able to develop molecular markers that are specific for the two herbicide-resistant AHAS variants using a cast marker system. And this system is PCR-based and utilizes the SNPs, or the SNP substitution between a wild-type ortholog and its corresponding herbicide AHAS gene, and also neighboring SNPs that distinguish the different, or the three different ALS orthologs. And this marker system will be a useful tool for the development of future improved herbicide-resistant Camelina cultivars at Smart Earth Camelina. The preparations for the commercialization of SES 1154HR already started when we had identified good tolerance to thiophansulforon in the original double mutant lines in 2015. But as you may guess, things often take a lot longer than you think, and there were a lot of pieces that needed to be put together. As such, Smart Earth Camelina Corporation obtained Canadian patent filing in 2019, and importantly, CFIA PBRA unconfined release, CFIA Animal Feeds Division, and Health Canada novel food approvals in March of this year. And this particular approval process was, let's say, arduous. And Jack and Debbie from Smart Earth Camelina can attest to that. The user requested minor use label extension, also funnily called ORMule, for the chemical Pinnacle SG Toss and Go, followed quickly after in April of this year, and plant breeders' rights were granted just last month. Certified seed of SES 1154HR was produced this year in Saskatchewan on about 180 acres, and the commercial production is slated for next year. Look out for the trademark New Gold, as this is what SES 1154HR will be marketed as. In summary, it took 12 years from mutagenesis to the commercial release, which is an extremely long time. Growers in Canada and the US have called for and anticipated a herbicide-resistant cultivar for a long time, and we are really happy to have finally gotten here. And with that, I would like to thank you for your attention and would be happy to take any questions.